Hello and welcome to the eOrganic webinar on late blight control in your organic garden. My name is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. We're a community of cooperative extension service personnel, researchers, ag professionals, organic certifiers, and practitioners. You can find all of our recorded webinars, information about upcoming webinars, as well as articles and videos on organic farming on our website at www.extension.org/organic_production. All the information published by eOrganic is about certified organic farming, but on eextension.org you can also find information for home gardeners. This is a special presentation specifically for home gardeners, since home gardeners can play an important role in minimizing the spread and impact of late blight on farms and gardens. In this webinar, you'll learn where and how late blight occurs, about the late blight disease cycle, how to look for and diagnose the disease, and how to manage late blight in your organic garden. I'd like to briefly introduce our speaker. We're very excited to have Meg McGrath back with us today. Meg, along with Sally Miller of Ohio State University, has already presented 2 webinars on late blight for organic farmers, which are available on our website. Sally was unable to join us today, but she contributed many of the slides in this presentation, so I've included her picture here. Meg McGrath is an associate professor of plant pathology and plant microbiology at the Cornell University Long Island Horticultural Research and Extension Center. Her research focuses on the management of vegetable diseases on commercial farms. After the 45-minute presentation, you will have the chance to ask questions. If you have a question, you can simply type it into the question box on your screen and hit return. If you can't see the question box, click the small plus sign next to the word question on your control panel to open it up. We'll answer as many questions as we have time for in the 30-minute question period. We're also going to ask you a total of 6 quick poll questions that will appear on your screen for about 20 seconds each about whether you've had late blight and how informative this webinar has been for you, or whether you plan to change any of your gardening practices as a result. Okay, now I'm going to switch control of the screen over to you, Meg.
Thank you, Alice. It's a pleasure to be here. Okay, the topics that I'm going to be covering this evening are the importance of late blight, diagnosis, pathogen biology, occurrence, and management. First, late blight is arguably the most important disease that occurs. It's extremely destructive. It can not controlled. It can wipe out a garden, a crop, needs to be managed. Other diseases that we have we can live with, tolerate their occurrence. This one is extremely destructive. It has to be managed. It's difficult to manage after onset. It's a very highly contagious disease, which means that if it's occurring in your garden and you don't manage it, the pathogen can move to other gardens, other farms, and they will be impacted. We consider it a community disease because of these points, because it is so destructive, so highly contagious. We all need to manage it together. We all need to know where it is. Here's some typical pictures of what late blight looks like. We'll show some more in a little bit. Top side of the leaf, a large lesion typically, and then the pathogen producing its spores on the lower side of the leaf. Here's an example of just how destructive it can be. This is an organic farm near me on Long Island. When the symptoms were first seen last year, the end of August, you can see the planting was just reaching the point of having fruit starting to come into production. When the farmer saw symptoms, here are the symptoms that we're seeing, typical large leaf symptoms, fruit symptoms already at that point in time. And this is what some of the plants looked like. Quite a few symptoms on it. And remember, this is the first time the farmer noticed that there were symptoms out there, already quite severe. It had been 10 days since the last time he'd sprayed copper. He did try to save this planting. He was unable to do it. It illustrates just how destructive the disease can be. So hopefully you now understand how important late blight is, that it's really critical disease to understand and to manage. And we'll move on to our very first poll question. You'll have about 20 seconds to let us know if late blight has occurred in your garden, whether it's been every year, whether you've seen it already this year, only had it last year, some other years as well, or you've never had it. And we'll share all these results with you because I think it's very helpful to have a picture of what we're all seeing. I personally had it in my garden last year. So it looks like most people have never had it yet. Very interesting, 49%, but quite a high number, 28% only had it last year. Diagnosis is an extremely important step with any disease to know whether or not indeed that you have that particular disease and understanding what other diseases and disorders can occur that look like it. Late blight affects the leaves, causes these typically very large lesions. Also very typical is this border of light, almost a light greenish wilted tissue around the brown lesion. On the stems, you can have dark tissue, as in with the stem in the background. Sometimes you'll get the pathogen actually growing right on the stem, which is the white that you're seeing here. And it can also hit fruit, a very firm, dark symptom on the fruit. And sometimes on the fruit, the pathogen will also sporulate. And that's what that is. Those are the spores of the pathogen. The typical signs of the pathogen, which is Phytophthora infestans, is this fuzzy growth on the underside of the leaf. So that's what you want to look for. If you see a symptom on the leaf, look to see if you see that. Now, if you had a really good hand lens or you had a microscope, this is what you would see. And if you had a compound microscope and you looked at these individual little balls that are here, you'd see they're actually lemon-shaped, and this is what it looks like enlarged. That's the pathogen. If you can see that, you absolutely know what you've got. There are several diseases and disorders that can kind of imitate late blight, and these are some of the things that have come into my lab and other diagnostic labs that people have thought maybe there was late blight and were not. So there's quite a few things that can occur and be mistaken for late blight, and I'm going to show you a number of pictures of these. This webinar will be posted, so you can go back and look at these pictures later. I have a lot of these pictures posted at my own website, and you can find a lot of pictures now on the web. So a lot of places you can go to get some help in initial diagnosis of whether or not you've got late blight occurring in your home garden. Initially, the symptoms of late blight can be very small little lesions. Now, this is a plant that was inoculated. This is just 48 to 72 hours after that inoculation occurred. So you can see you can get symptoms pretty darn early, quick. Very unusual to catch it quite this early in your garden, but it could happen. You could see very early symptoms. More typically, you're going to see the larger leaf spots. Here's some fairly early ones on tomatoes with some wilted tissue there, the more typical symptom we saw earlier. Sometimes it can be more of a brown look. And if you don't see the pathogen present, you can take these leaves, as I did, put them in a plastic bag overnight, and you can see the pathogen developing. If it takes more than a day, you're probably looking at something else. But just overnight could be the pathogen. Drought stress is probably the most common mistaken symptom for late blight because you get these large patches of dead tissue on the leaves. And clearly in this situation, big plant, little pot, the plant's just not going to be able to take up enough water and satisfy those leaves. And here's a close-up of what they look like. And what you would want to do if you had these kind of symptoms in your garden and you wondered, gee, could it be late blight? Is look around for other symptoms. Do you see anything on the stems? Do you see anything on the fruit? And look at the underside of the leaves. You're not going to see the pathogen present on the underside of these leaves. Here's another picture of the drought stress. It's going to be along the edge of the leaves, no spots in the center. There's another characteristic of it. And you're seeing more of a yellowing of the border, not that wilted look that we saw, that light green look that we saw with late blight. Another picture of drought stress. In this situation, there's not much of a border at all, just a sharp line between the affected tissue and the healthy green tissue. You would not see that with late blight. You'd see some kind of a border here typically. And here's that late blight symptom again. Notice the border, that nice light border. And then on the underside of the leaves, you're seeing the pathogen sporulating, which you would not see with drought stress. Gray mold, another common one confused for late blight. I'd say this is number 2 on the list of samples that have come in to me. You can get big lesions with a gray mold that kind of look like late blight. A little bit of a concentric look to them, more of a yellow bordering, not the same kind of bordering that we saw with late blight. And you typically will see symptoms elsewhere on the plant. So here we're seeing some fruit symptoms. We're seeing flowers that have died back due to gray mold. And the characteristic fuzz of gray mold, it's a more grayish shade. It's not the white of late blight. And the fruit is, you could just kind of guess that that fruit is soft, whereas it would not be with late blight, and it would be more of a brown discoloration. So that's how you can distinguish those. Septorial leaf spot is probably 3rd most common that's come in to me. Does cause spots on the leaves, but they're much smaller spots. And the characteristic with this disease is a tan, very light colored center to the spots. And if you look really close, you can see it here where you see a little bit of a white, the pathogen oozing out of these lesions, but much smaller. They're all much smaller. Can also be on the stem, much smaller spots than you would see with late blight. And here are the lesions on the stems with late blight. As I said, a larger symptom. The sporulation is white, a very white look when it occurs. Here's a very early start to the stem lesion occurring here on potato. Here is a very early symptom on a tomato very young seedling where it's a kind of a discolored look due to late blight. Here it is on stems in the field. Because these stems are more firm, you've got more of a dark brown look to the lesion when it's on field plants. Here's Botrytis gray mold again, and you can see it's a softer look on these plants and fuzzy growth. If you were to look up close at these plants, you'd see more of a fuzz. It's a thicker growth, and it's more gray. It's not the white. And typically with gray mold is it starts on dead tissue. So here is a leaf that's dead. The pathogen, which is called Botrytis, was able to grow on this leaf, and then it got established on that dead leaf, and then it was able to attack the stem of the leaf, of the plant itself. So that's another characteristic to look at with Botrytis, is that the gray mold is going to be associated with dead tissue, not just developing on a healthy stem, as occurs with late blight. Here's another example of Botrytis gray mold that came into my lab this spring. And you can see with this case, that kind of looks a lot like late blight. But as you look further up on the stem, there's the growth of the gray mold. It's a very different look, at least for my trained eye as a diagnostician, a very different look than with late blight. And also with that sample, if you get a little closer look, you can see it's very raised. It's a much higher growth due to Botrytis gray mold than you would see with late blight, more of a fuzzy look. And commonly, as I mentioned before, coming in off of dead tissue. So you can see a dead leaf. There's the gray mold, much wider growth, sticking much further off of the plant tissue than you would ever see with late blight. Another sample that came in, this was an injury due to a chemical. Looks a lot like late blight, but there were no leaf symptoms on this plant. Looked a little bit subtly different to me, at least, from late blight. Here's a close-up to show some of that injury. Not quite the same as late blight, but as you can see, it's quite similar. The grower who had this happen was very concerned, thought he might have late blight. We were able to run a test on it and prove that it was not. So here to contrast is symptoms of late blight on the stem, injury. Now, to someone who doesn't have the training I do, that would probably be a very difficult call. And it's a situation like this. If this was my plant and I didn't have the training that I have, I would want to take it into a clinic to get it diagnosed. Hard to tell apart. Late blight can cause dying back of the top of seedlings. This is all late blight. Some leaves that have been completely blighted out. There's a virus that can attack tomatoes called Tospovirus. Can cause the same type of dieback. But if you were to look around on this plant, you would see other kinds of symptoms that are unlike late blight. The fruit does have a firm brown discoloration, but there's more circles here definite, almost a ring spot that's occurring, which you will not see with late blight. And if you were to look at some of the leaves, you'd see much smaller circular lesions occurring on the leaves, very different from late blight. So take-home message is to look at your plant, look at for other kinds of symptoms on a plant. Don't just stop when you see the first symptom, something on the stems or a dieback of the top. Look around, see what other symptoms you've got to try and finish the diagnosis. Late blight can also affect the fruit. They're causing this brown, often starts at the top of the fruit I've seen with plums. Can be anywhere on the fruit, more with your round type, beefsteak type tomatoes. Cherries, I often see the youngest fruit getting hit first. You can see the brown discoloration on those. And it tends, at least initially, to be more of a firm rot on these fruit. Here's the Botrytis gray mold. You can see the growth up close. It looks a lot different from late blight. And notice the fruit. It's more of a soft rot. There's no brown there. Help you to distinguish those two. Here's another example that came into the lab of Botrytis gray mold, and you can see the fruit does look discolored, so there was a lot of concern that this might be late blight, but you can see that that fruit is soft, not like what you would see with late blight. And this particular one, we ran a test kit which came out negative. So there are ways that we can test it in the lab. Blossom end rot has been one that's been confused for late blight because you can see symptoms up on the fruit that are somewhat brown, as you do with late blight, sometimes not at the blossom end, which is characteristic, so there would be more characteristic blossom end rot where it's bright, dark, very black at the blossom end. I've had some situations where it's more of a brown look. But you can see that fruit is kind of soft, and that's not what you're going to see with late blight, at least especially with the initial symptoms. It will be brown and firm for a while. Early blight's another common disease of tomato. Very small leaf spots compared to late blight, and the symptoms on the fruit are quite different from what you see with late blight. There are several bacterial diseases. I'm showing two here. Bacterial soft rot, which can hit the stems. With this disease, you wouldn't be seeing any symptoms on the leaves, one way to distinguish it from late blight. And bacterial canker, much more of an edge look to the symptoms. There's also a bacterial speck and spot that create very small spots on the leaves, very different from late blight. Now, if you think you've got late blight, you definitely want to get it diagnosed. There are several diagnostic labs. Every state has a diagnostic lab that you want to take your sample to. And you want to do this not only to confirm and know that you've got late blight or don't have late blight, but by the lab knowing where late blight is, they can notify other gardeners and farmers. And this gets back to the fact that this is a community disease, and we need other people to know that late blight's occurring so that they can react. So very important. If you think you've got late blight, take it to a lab, get it confirmed. Or even if you're quite sure you've got late blight, get it identified, confirmed, so the notification process can get started. So where can you find a diagnostic lab? You can go on the internet for the National Plant Diagnostic Network and go to the different areas, so different regions, or you can click on the map here for the different regions of the U.S., depending on where you're located, and find a diagnostic laboratory. Hopefully you can take your sample directly to the lab, because that's the best. You've got a good quality sample. You want to make sure you pick a good sample. If your plant looks like this, it's really dead, it's going to be very difficult to tell what happened. So having a good sample, representative of what you're seeing, it's got to be in good condition. You either want to take it there yourself or consider sending it by overnight mail. Don't rely on regular mail. The sample arrives, if you send it in good shape but it arrives in horrible shape, the diagnostician is not going to be able to tell what happened. And it's also good to provide information. That can be really helpful in completing the diagnosis. So filling out your form completely is very helpful. And in the lab, what are they going to do? They're going to have a microscope. They can look up close, and they'll be able to see the pathogen and see the characteristic spores. With this pathogen, it produces a lemon. That's a very lemon-shaped spore, very characteristic. How it sits on the leaf, these structures that support the spores, very characteristic. And somebody who's trained in a laboratory will be able to make that diagnosis. There's also diagnostic kits. You could purchase these yourself on the internet, but better to take it to a laboratory, let them run the test, because then they know and they can move on to the next step of notifying other people where it's occurring. So here's two examples of the test kit. Here's one where there's only one single blue bar for the control, and here's a positive sample where you can see there's a blue bar for the test sample. Here's the sample in the bag. You can see characteristic symptoms of late blight. So you can see test kit, it's very quick. It's very similar to some of the other immuno tests that are out there these days, such as the pregnancy test. Very quick, accurate results. Very helpful. It's important to understand the biology of any disease and the pathogen that's causing it. Having an understanding of the pathogen and the disease helps an awful lot when it comes to managing it. With this particular disease, it's caused by a pathogen whose name is Phytophthora infestans. It's important to understand that it's an obligate pathogen. That means that it needs living host tissue, plant tissue, in order to survive. It can also survive as a structure in the soil, but we don't believe that that is yet happening in the United States, and I'll get into that in a little bit more detail. But at this point in time, our understanding is that it needs living plant tissue in the U.S. to survive. It also has a narrow host range, and that's a very important fact when it comes to management. Here's the life cycle, and there's a lot of life cycles that are posted on the web now for this pathogen. I got this one from this particular site. With our fungal pathogens, they have an asexual cycle, and some do or don't have a sexual cycle. So it's very different from mammals, which we're used to thinking about, or from plants. In most of our pathogens, their primary cycle is this asexual one. And we believe that this is the only cycle that is occurring in the United States at this point in time. Important to recognize it has this other cycle, however, that it could be reproducing sexually. So what typically happens is it survives on living plant tissue. Potato tuber is a living plant tissue, so if there's a potato tuber that's left in the ground, in a coal pile, used to plant a potato crop or in a garden, that can be a primary, and that's been a very important source of the pathogen. Because the pathogen has survived in this potato tuber. It's then able to grow and get up into the leaf tissue as the plant grows. It starts producing its spores on the leaves. So there's its spores up close. There's a cross-section of a leaf with the spores and the structure that holds the spores on the underside of the leaf. So that would be, say, right here on the plant. So it's producing spores. And it doesn't take it long. It's about every 4 to 10 days that the pathogen goes from the spores on the leaf to being dispersed in the air, to landing on another plant, to having a lesion producing spores. So this is a very quick cycle, and it just keeps going. And the pathogen is just reproducing itself over and over and over again. So it's the same individual coming out over and over and over again. And actually, each of those little lemon structures has many little spores inside of it that can develop. So it's got a couple different ways it can germinate. But that's getting probably a little bit more into the biology than you need to understand at this point in time. The other cycle is the sexual cycle. And that, just like with us, takes two different individuals growing together. And then what happens is the pathogen can produce a spore type that can survive in the soil over winter. Then it germinates, and the cycle occurs all over again. So you've got these two cycles that can occur with the late blight pathogen, and with a number of other fungi do the same thing. So, key fact is that you've got two mating types with the pathogen Phytophthora infestans, and this is true with some other fungal pathogens. Since the pathogen doesn't produce structures that distinguish it like males and females, we call them A1 and A2 instead of calling them males and females. So remember that with pathogens. They're A1 and A2 mating type. Typically in the U.S., what we've had when late blight has occurred is we've had what we call a clonal population. That means the population is all one individual continuing to multiply itself. So it's all genetically the same, which also means it's all one mating type. And if you have just one mating type, be it the A1 or the A2, the pathogen can only reproduce asexually. And that means you get these spores produced, those are the sporangia, and they're very short-lived. They can't survive long. and they are affected by UV radiation. So if it's a sunny day, those spores can't move up into the air and be carried any distance. They're going to be killed by the UV radiation. So short-lived. Particularly, they'll move around on a rainy kind of a day, an overcast day. If both mating types occur together, which means they're infecting the same leaf or stem and they grow together, they can reproduce sexually. And that occurs right away, pretty much, as soon as they've grown together. And what happens is it produces what's called an oospore, and that is long-lived, and it can survive in the soil. So you can see, here's that oospore. You can see its thick wall compared to the thin wall on the sporangia or sporangium. That spore is going to be able to survive a long time. It can be in the soil, doesn't need a living host. It won't grow until and multiply until it finds a host to grow. But it can survive, and it can survive for years. And that's an important thing to keep in mind. So typically, here we have those sporangia forming. They're on the underside of the leaves. That's all that white. Those are those wind-dispersed asexual spores. There's a lot of them. So in this circular area, there's 100,000 to 300,000 of those sporangia. That's a lot of spores. And each one of those spores could be picked up by the breeze and moved on to another garden or farm. Typically, they don't move too far, about 20 miles maximum. Also, the cycle can be pretty quick, 4 to 10 days from that spore moving, landing on another leaf, producing a new spot like this, and producing more spores. So very fast growth and development and spread with this pathogen, which can be very devastating. So typically moves up to about 20 miles, but you can get some further long-distance movement. Host range, pretty narrow, which is fortunate, but important to realize. Our primary crops have been tomato and potato. Some strains do go to tomatillo, but the strains we had the past year do not appear to be going to tomatillo, which is good news. But we do have different strains that can go to different hosts, and that's important to realize. We had a strain a couple of years ago that started on petunia and then moved from there to other crops. And we've got a number of weeds and other ornamental plants that can be important hosts of the pathogen. And you'll notice that most of them, these are all in that solanaceous family. So it's all related plants. So thank goodness the host range is narrow. That helps us a lot in managing. Another important fact to realize is that strains can differ in their virulence. Typically, the pathogen strains that we've had really attack potato. But last year, we had strains that were very aggressive on tomato and caused very few symptoms on potato. So we had situations where a planting could be very badly hit with late blight in tomatoes, as in this picture, potatoes adjacent nearby that had very few symptoms. Occurrence in the United States. To understand the occurrence the past few years with late blight, we need to understand the basic requirements for a disease to occur, because this determines when and how and why late blight occurs. So typically, and this is true, these are the basic requirements for any disease to occur. You've got to have a susceptible host, and in the United States, we've got a lot of susceptible hosts because a lot of us grow tomatoes and potatoes, a lot of gardeners, a lot of farmers. Host is grown throughout the United States. We often have favorable conditions, but not always. So that would be the second determining factor. And the pathogen you'll see I have in the smallest letters because that tends to be the limiting factor with late blight. The pathogen most often is not present. So what are our sources of inoculum? In the United States, our primary source has been infested potato tubers. It's an obligate pathogen. It can survive on potatoes and be in the soil and come up in the spring. Those potato tubers, if they survive the winter and they grow, pathogens can be infected. The pathogen can be growing on that plant tissue, and that can be the start of disease development. The pathogen can also survive in frost-free areas. So for instance, Florida, southern parts of Florida, most years don't see a frost. If there are tomatoes there, the pathogen can survive there and continue to develop. From those plants in frost-free areas, there can be wind-dispersed spores, which then move and could move to other areas. So if it's occurring in Florida, occurring into the spring in Florida, now you've got plants being grown in Georgia or South Carolina. Slowly but surely, the pathogen can move up the eastern coast each year. Infected tomato transplants have occasionally been a source for the disease. It was a major source last year. In previous years, it's been much more of a minor source. But there's been situations where plants have been purchased by commercial growers that were already infected, and then last year a number of gardeners purchased plants that were already infected. And then there was one year that we had petunia plants that were infected, and that was an important source of the late blight pathogen. It's important to realize that there is another source in Europe that's been occurring in recent years, and that is oospores. The pathogen has started to sexually reproduce in Europe. A number of years back, they introduced a second mating type, and that mating, now they have both mating types there, so the pathogen is able to sexually reproduce. And that has really changed late blight occurrence in Europe, and I think that's something very important for us in the United States to realize, that if the pathogen does start to reproduce sexually and produce these oospores and is able to survive other than in potato tubers or frost-free areas, we will have different occurrences of late blight in the United States. Typically, we have those asexual spores, and they move short distances. So here are the asexual spores on the underside of the leaf. They will move up to about 20 miles typically, but occasionally they can move further. And we had an outbreak on Long Island, where I'm located, back in 2007. We saw symptoms at this commercial field that I took pictures of on the 26th of August. The nearest known occurrence of late blight that year was a report out of Pennsylvania the 10th of August. So we think the pathogen made that huge jump that year. Doesn't often happen, considered a rarity, but it is capable of moving that far, and I think that's important for us all to realize that that can happen. And this particular photo I've included of some of the fruit from one of his other fields, where it was a very subtle symptom on the top side of the fruit, and didn't really notice it until after he'd harvested it and got it into the bin. So it was quite a loss for this grower. And you can see these fruits, these plants, and the fruit on these plants, just how much devastation in this other planting of his. Very, very destructive pathogen, very destructive disease, can cause quite a lot of losses. So occurrence in the United States recently has been occurring in Florida every spring since at least 1993, and since 2005, late blight has been developing into May. That's a really important fact to understand. The pathogen, we think, has developed some new strains that are able to tolerate warm temperatures, able to survive into the spring later than it used to survive into maybe March. Now that it's present in Florida into May, it means there are more hosts further north. So we now have more of a green bridge for the pathogen to move up the coast than we've ever had in the past. In Maine, we've had late blight occurring every summer recently in the major potato production area, which is the northern part of Maine, and also into Canada. And that's because of the pathogen being able to survive on potato tubers. Elsewhere, late blight has been pretty sporadic, and it's been pretty rare, thank goodness. In potato, here's some dates of first detections in the potato production area. So you can see every year it's occurred for the past few years, and often occurring about the same time. And that's reflecting the fact that it's in the potato tubers, and it's taking just the right kind of environmental conditions for the pathogen to start developing and get to a point where there's symptoms present and we can see them. Whereas where I am on Long Island, I've been here since 1988 until this year. I'd only seen late blight 5 times. I've seen it once now this year, so I've seen it 6 times in my career here on Long Island. And you can see it's all been in fairly recent times, and it's been very erratic when its first date was. You know, one year we had it as early as July, many years not until October or late in August. And that's reflecting the pathogen I think, just slowly moving up the eastern seaboard until it finally makes it up to Long Island. Last year was the earliest we had seen it, until this year when we saw it a little bit earlier in June. So we've got some local sources of inoculum now that we are starting to see it earlier. So the situation is changing once again. Last year, we had several strains of the pathogen which are unique. We had our usual potato strain. That would be that one clonal population that's occurring over and over and over again, one mating type. But we also in tomato had several different what we call genotypes, mostly new genotypes occurring. What was a little bit alarming is that we did pick up both mating types in some areas, only the A2 mating type here in the Northeast where I am, but we did pick up the second mating type, which would be the A1 mating type in Virginia and Pennsylvania. We do not believe at this point in time the pathogen is sexually reproducing anywhere, including Florida, where both mating types have also been found. But it's important to realize that both mating types have been found in the U.S. That possibility is there. So this past year, when has late blight occurred? We had it in Florida, January through May, similar to some past years. Also, they had some pretty nasty freezes. It did not completely kill all the tomatoes back in January, so late blight was able to survive. Louisiana had some outbreaks early on, and then up in the greater Northeast, we started having some outbreaks beginning in, the grower thought it was April. This is the farm. This is the situation of it being in a high tunnel. We had some greenhouses, gardens. We've had one occurrence in New York so far. That was on Long Island. That was a garden. Some more gardens in Connecticut. And then we get into late May, late June rather, and then into July. We're starting to see some commercial fields with it, but still mostly we're seeing gardens having outbreaks. And very few outbreaks this year. Conditions have not been as favorable, thank goodness, as last year. That's helped a lot. And there hasn't been as much inoculum, I don't think, surviving over winter as we feared. So that's all good news. So today it's mostly been tomatoes that have had late blight. We have had potatoes, but mostly tomatoes. We've had greenhouse occurrences. We've had high tunnels. Had a situation with retail stores having affected plants. That was mostly Manitoba. Had a situation similar to what we had last year. Several gardens and a couple commercial fields. What we think has been the source in the northern states is infested potato tubers, but we haven't been able to point our finger at any particular field as being the source, or plants in a garden as being the source. Part of it is that it's hard to find that smoking potato gun, so to speak, to find the actual source of an outbreak. A colleague of mine who works on late blight sees it every year. He works on it up in Maine, says he's very rarely actually found the source of late blight outbreaks. I think another thing that's critical to realize, because it is hard to find that source, and that's where you as gardeners can be great help to us, that if late blight occurs in your garden, helping us determine where it came from. Did it come from potatoes, or did it blow into your garden from another planting? That's where we need to definitely work together to try and understand how the pathogen is surviving, where is it coming from. So another reason to make sure you get information about how you're planting, where the late blight might be coming from, get a confirmed diagnosis of late blight. Managing late blight. Several steps to managing late blight. First and foremost is to select resistant varieties. At this point, there are not varieties that are available that have been bred with specific resistance genes to late blight. They are coming. There's a lot of work going on to develop resistant varieties. They're not yet commercially available. Last year, a number of gardeners and also commercial farmers observed tolerance with a couple of different tomatoes, in particular Matt's wild cherry, some other cherry types. There's some other varieties that looked like they had some tolerance. So they certainly are an option, one way to try to control late blight. But there are some varieties coming, and two in particular I'm showing here, Mountain Magic and Plum Regal. These were developed by North Carolina State University, also input from Cornell University. There's some other universities, Penn State, that are working on resistant varieties. There are other seed companies, public breeders, Johnny Seeds is working on resistant varieties. So there's going to be a number of sources coming. Right now, unfortunately, not commercially available this year, but hopefully next year we will have some varieties available for you to grow. Step number 2, and probably the most critical, is to start with clean potato seed if you're growing potatoes in your garden. You want to have certified potato seed. Ask when you buy your seed. You don't want to use table stock that you can get from a grocery store. There's a higher tolerance for pathogens and for disease in potatoes that are being sold to be consumed than there are in potato that's being sold as seed. So make sure you start with good seed. And that also means don't save potato seed from your garden to use to plant the next year. Here's showing what an infested potato looks like, a tuber looks like. You can see some of the symptoms in that potato tuber. Here's the plant that grew up out of that infested seed, and you can see some symptoms of late blight on that young plant that has grown up. Next step is you want to destroy any what we call cull potatoes. So that would be potatoes that you had that you kept from last year that started to break down in your refrigerator or your other storage area. Don't just dump them out on your compost pile. You want to destroy them. You can bury them very deeply. You can put them in the trash. You don't want to have a pile where you've got infested potatoes. The reason for that is the plants can grow from here and produce these symptoms and have the spores and spread from there. And this has been one of the primary sources of the late blight pathogen in some of the potato growing areas. And growers have learned this is very important to manage these. They do a much better job these days than many years back when they were first learning how to manage late blight. And now that it's affecting more of us gardeners, we need to learn as well the importance of using good potato seed and destroying those cull potatoes. And next is you want to make sure you've destroyed any tomato and potato volunteers that grow. So here's a volunteer coming up of a potato. And you also want to control and destroy any susceptible weeds so that you don't have a source of the pathogen and you don't have a place where the pathogen can multiply and produce spores that can move over to your crop and in your garden. So tomatoes produce an awful lot of seed that can come up the next year as volunteers. So this is my own picture from my own garden, my peas, and there's a little tomato coming up. You want to control that volunteer, not because it could harbor the pathogen when it started to grow. It can't be in the seed. It doesn't have the pathogen can't survive in tomato seed. But that's a weed growing there in my peas. I'm not going to take care of that plant. If late blight started to develop on there, I might not see it, I might not manage it, and then that could be a source of the pathogen then moving over to my tomato plant. That's why it's important to control tomato volunteers. If you have tomato volunteers growing in your garden that you are going to take care of, I know a lot of people like to let their cherries grow from year to year. That's fine. If you're going to take care of that plant and treat it like a crop plant, it's no longer a volunteer. You're going to take care of it. If late blight develops, you're going to see it, you're going to manage it. It's an entirely different situation. You also need to control weeds that can be alternate hosts. And a very important one that appeared last year was Bittersweet Nightshade. It had symptoms of late blight. This is a plant actually that came from right near my garden. These are the typical symptoms of late blight on this weed, very large lesions, just like you see on tomatoes and potatoes. Here's a close-up of those symptoms, those large lesions. And I was able to see the pathogen right on the underside of these leaves, confirming the diagnosis that indeed these lesions were caused by the late blight pathogen. Continuing on in management, the next step is to inspect those plants regularly and thoroughly. You want to be out there at least every week looking for symptoms of late blight. You want to look carefully throughout the plant. If you see affected tissue, you want to destroy it. First and foremost, make sure that it is indeed late blight. So you want to make sure you get a proper diagnosis. There's so many imitators that can fool you and look like late blight. I've had a number of gardeners this year that have been nervous when they saw drought stress or saw Botrytis gray mold and thought they had late blight. Make sure you got it properly diagnosed, but don't delay getting that tissue destroyed if indeed you've got late blight. Next step in management is to apply fungicides. This is a very difficult disease to control by just removing plant tissue. Really your only other option for management. If you've got a good resistant variety, you'll have a lot less affected tissue. You might be able to slow it down that way. I spoke with a gardener who spent a lot of days last year trying to manage late blight in his garden by just removing affected tissue, and he eventually lost the plants to late blight. So you can slow it down for a while, but it's not, unfortunately, going to solve and completely manage late blight. If you're going to use fungicides to manage your diseases, first, there are several out there. What you need to realize is that efficacy is not a requirement for a fungicide to be registered in the United States. Fungicide and other pesticides, the registration is done by the Environmental Protection Agency. The focus is on the safety of the product and what happens to the product in the environment, impact on non-target organisms, how quickly it breaks down. Efficacy is not considered. Important fact. Copper has been one of the ones that has shown the best efficacy. There hasn't been a lot of work looking at products available for organic growers and gardeners. There are a number of other things out there. At this point, copper does appear to be the most effective. It's important to realize when you're managing diseases with fungicides that a diseased plant cannot be cured. You can't apply any fungicide and get rid of the spots that are already here on these leaves. It's not like when we get the flu and get pretty sick and we can go to the doctor and he can make us better. A diseased plant's entirely different. So our focus is very much on preventing the initial infections from occurring rather than trying to catch up and save the plant, particularly with late blight. It's just very difficult to slow it down once it's already started. Some other diseases like septorial leaf spot and early blight, you can wait until you see symptoms and then start applying an organic fungicide and slow the disease progress. Not always the case with late blight. In fact, usually not the case. Other thing to realize when you're using fungicides is that there are safety precautions to be aware of, particularly for the applicator. You want to look at the full label. Don't just read the part of the label about how much you need to apply. Make sure you know what the potential hazards are of the product that you're using. It's usually as you're actually applying it, not later on. With copper, you will see on the label that it can cause eye damage if it gets into your eyes. You absolutely want to be wearing goggles. Whatever I'm spraying, I wear coveralls, I wear goggles, I protect myself because I don't want to take any chances with what I'm using. And you should consider the same. And be aware of what those precautions are as you're considering what you're going to use. Maybe that will make you decide that you don't want to use that particular fungicide. Better to be aware of it and know it up front. So that's part of your preparing. If you're going to use fungicides, prepare for it. One thing, you want to make sure your plants are well enough spaced in your garden so you can get in and get good coverage. Know what you're going to use. Do you have the precautionary equipment that you need? Do you have eye goggles to protect yourself? Plant your garden such that when you spray your tomatoes, you're not going to be spraying anything else that you don't want to have hit with spray. I've often put lettuce at the tail end of my my planting in my garden, and that's a consideration. Do I want to be spraying copper and having it land on my lettuce or not? That might dictate how you want to organize your garden. Important things to think about. When to apply fungicides. With late blight, you really need to apply them before the pathogen has started to infect, before you've got your first infection out there. So it's different from your other diseases. It's not a disease you can live with. It is so destructive, it's one you just can't tolerate. Other disease, it's the only disease I personally will try to control in my garden. Other things, I can accept losing some fruit. That's okay by me, but late blight is just so destructive, you're likely to lose everything. The pathogen has to be present, and you have to have favorable conditions in order for late blight to develop. So you also want to know, is the pathogen around, are conditions favorable, and that will help you decide whether or not you need to use the fungicide. So monitor reports of late blight. That will help you to know where it's occurring, whether or not it's a concern this year. There are reports up on the web. I have a link at a website that I have where I've got a lot of pictures and information about late blight. It's on Google Docs. It is being posted by anybody who feels like posting a report, so there may be some that get missed, but number of reports went up so far from Florida and then Louisiana. You can see them there. Sometimes they are confirmed, sometimes not. So if you see here that it was done by a lab, you can feel pretty comfortable, or a microscope, that it is a good diagnosis. That's an important consideration. So late blight reports are up on the web. Hopefully we'll get more communication going amongst gardeners as well in other ways as late blight occurs each year, so we all know where it's happening. And that's an important part. If late blight occurs in your garden, you want to make sure your neighbors know about it as well. The Cornell Integrated Pest Management site maintains a weekly late blight update, and that would be for New York and the states nearby. Here's an example of some posting. I like to show this one because it illustrates that no amount of late blight is too little to report. So here's a report back in 2006 of a single late blight lesion having been found. That's how important this disease is with farmers. When it occurs, we get the word right out to farmers and let them know what's happening, and we share the information with other extension staff. It's the only disease that we have an update and a listserv where we're getting information out to each other. That's how important this disease is. And if you were to scroll down that page, here are the other reports of late blight in 2006. You can see very few reports. That's a typical late blight year. We don't have a lot of late blight occurring in the greater Northeast. So after looking to see if there's reports in your area and the pathogen's present, if the pathogen is present, the next thing you want to know is, are conditions favorable for late blight? Rain provides very favorable conditions, but also high humidity can be enough with this pathogen. There is a forecasting site, and I have it linked at my website. And here is that forecasting website that you can go to, and this is just for the northeastern U.S. There's another forecasting system that's occurring up in Maine. There's some other more local ones that are out there. But this one is using various weather sites that are located around the Northeast and predicting when conditions are low for disease, when spores might be forming, and when infection is possible. So that's an important site to look at. But realize it's not considering whether or not inoculum is present, only whether or not conditions are favorable. Pathogen's not present, late blight's not going to occur. Here's that field again where we had the outbreak back in 2007. During August, we'd had two major rain events prior to when that late blight sighting occurred back in the end of August. And temperatures had been fairly warm, typical August weather for us on Long Island. So it doesn't feel like late blight weather, but there was high enough humidity, and those two rain events were enough. That's something important to keep in mind. It isn't rainy every day weather that's needed for late blight. Here's that field I showed you earlier back the end of August last year where the severe outbreak occurred. If you look at the weather prior to that outbreak in August, we hadn't even had an inch of rain all of August. Similarly, fairly warm day temperatures that last August. Those are the conditions before late blight was seen the end of August in that planting. But if you were to go to that forecasting website on the previous Wednesday, the pictures I showed you on the 31st, that was a Monday, so a little less than a week prior, we had a lot of favorable conditions, and that red dot right there of infection possible is right around the location of where that farm was. So this forecasting system actually did tell us that conditions were favorable when that infection occurred. So it's a very useful site for knowing whether or not you're going to need to apply fungicides. So you want to apply fungicides preventively when the risk is high, pathogen present, disease, the environmental conditions are favorable. And if you think you've got symptoms of late blight, it's really critical to get an accurate diagnosis. Take it to a diagnostic lab. Know whether or not you indeed have late blight occurring. You want to report your outbreaks. If you take it to a diagnostic lab, they will help with getting it out there. But if you've got other gardeners and you know you've got late blight, you want to make sure they know about it. We consider this a community disease. You need to share information, and that way other people can react and help save their planting from the spores that might have been moved out of your garden already. It's important to destroy late blight if it's not being managed in your garden. You want to do it promptly. The best time is to do it on a sunny day if you've got that option, but I wouldn't wait too many days to do it. The reason to do it on a sunny day is that as you pull those plants, you're going to disturb spores. They'll get up in the air, and the UV radiation will kill them. The spore is short-lived. It cannot survive sunny conditions. Cloudy day, it can get up in the air, and the clouds protect it from the UV radiation. What can you do? You can take those plants. You can bag them. You can have a pile and just cover it with a tarp. That works better for a grower or a gardener who's got a large planting. You can also bury them as long as it's good and deep. You can probably safely put the plants in a compost pile if they're deep. Don't want to put tomato tubers in there. One concern is whether or not the pathogen has sexually reproduced on your plant tissue and survival structures are present. If that's the case, then you really are better off bagging that tissue and getting it out of there, not having it survive and stay in your garden. We don't think it's sexually reproducing yet, but it could be. So here's a community garden on Long Island where late blight first outbroke this year. There is right here a planting of potatoes. We think that might have been the source. We have yet to be able to determine that. Here's showing a bag with some affected plants that are being properly disposed of. The reason to leave them in the bag in that sun is that they're going to heat up in that bag, and the plants are going to decompose, break down a lot faster than if that bag was just thrown immediately in the trash. Here are some of the last plants that hadn't yet been pulled up. Here's a close-up of some of those plants at their base. You can just see the amount of symptoms. This is less than a week from when the gardeners first saw symptoms in this garden. Came on pretty darn fast, illustrating just how destructive late blight can be, how quickly the symptoms can come on. These gardeners said somebody was out there in one of those community plots pretty much every day, so they knew that the symptoms had started a little bit less than 1 week prior to this point in time. But very destructive disease, very hard to manage. And I want to reiterate once again that at this point in time, the pathogen appears to be just reproducing asexually. That's a short-lived spore. There is the potential for it to reproduce sexually. We have detected both mating types in the United States. Very rarely. We usually detect just one. But if it does reproduce sexually, it produces a spore with a very thick wall that can survive long-term in the soil. At that point, late blight becomes like early blight and septoria leaf spot and other diseases where the pathogen can survive in our garden. So hopefully you've gotten the take-home message, which is that managing late blight is essential. We've got to have an aggressive management program. It's warranted to minimize the impact to other gardeners, to other farmers. If you don't manage late blight in your garden, you are putting other gardeners and farmers at risk of having late blight develop. And another reason to be really aggressive about managing and not just allow those plants to just sit in your garden for the rest of the season is if, by chance, both mating types did appear in your garden, the sooner you get rid of the plants, the less the opportunity there is for those two different mating types to grow together and oospores to form. Once that happens, the pathogen can survive right there in the soil. Very critical. We don't want that to happen. Another big challenge we've got is educating everyone. So we've reached a lot of you tonight, and hopefully you'll share what you've learned with other gardeners and help get the information out about how to manage late blight, and that we can't live with it. We've got to have a good management program. And hopefully we will soon return to late blight being a less common disease. I do expect we will continue to see it late in the season because it is developing later in the year in Florida. We've got that new source. But hopefully we'll have less occurrence of it surviving over winter and starting early in the season, which is when it's really devastating for all of us.
You can find the webinar recording and more late Blight articles at the address on the screen. That's eOrganic's website, which has lots of other articles and videos on organic farming. It also has all our archived webinars. And then if you have additional questions at the end of the webinar that you haven't got answered, or if you think of any more questions, you can go to http://extension.org/ask, and there's a free Ask an Expert service that is part of the National Online Extension Web Initiative, so you can get some of your questions answered that way. We'll also be sending you a follow-up survey, and we really need your feedback, so we'd really appreciate if you could fill that out if you've enjoyed this webinar so that we can keep offering more webinars in the future. There's also more information that Meg has posted in the form of a handout at the address on the bottom of the screen there. That's article 28624. Meg has posted one of her handouts to that page. Okay, so now we'll get to the questions. Let's see. Okay. What is the typical period of time that the late blight disease needs to occur? I know you mentioned that, but you might want to repeat that.
It can develop in as short as 4 days. So that's 4 days from a spore landing on a leaf, growing into the leaf, and a spot developing.
Okay.
And that's been with the more aggressive strains. A week is more typical.
Okay. Here's a question about whether late blight is aided by stressful growing conditions such as weather, humidity, or high temperatures.
Stress, not so much. It's more the conditions that are favorable for the pathogen. And that would be wet or high humidity that allows the spores to germinate and grow and get into the leaf. That's the critical thing. If it's really hot, it does slow the pathogen down, but the pathogen does tolerate warmer temperatures than it has in the past. 90, 100-degree weather, we're starting to get into conditions that aren't favorable for the pathogen.
Okay. Let's see. Someone wanted to know if the fruit is still edible if the plant has late blight.
That's a good question. What I would do is cut off the tissue that's diseased and consume it fresh. It may, because the tissue is going to start breaking down, it may have a slight off taste, just as you would have with any other pathogen. The late blight pathogen does not produce a toxin. It's not considered to be any more harmful than other pathogens that attack fruit, such as anthracnose. We often have spots on fruit due to anthracnose. I see that all the time in my home garden, and I try to salvage what fruit is left. I would not use a tomato that has any kind of disease symptom or even bruising to make food that I'm going to save, such as canned or even frozen. Particularly canned, what can happen with a fruit that's starting to spoil is it doesn't have low enough acidity, and that can isn't going to stay tight. It's not going to stay good. You won't have a good seal. I've had some gardeners who complained that commented that a couple of years back, when they had late blight, they had trouble with their cans not staying secure through the winter.
Okay, here's a question about potatoes. If infected potatoes overwinter and come up in the spring or early summer, will they look diseased right away?
Not necessarily. And you might see symptoms, you might not. One of the concerns has been that the strains that we had last year that were occurring on the tomatoes aren't as aggressive on potatoes. So the potatoes wouldn't have had as many symptoms, and we won't see as many symptoms this spring. So we were anticipating that more tubers with late blight would survive in the ground because the pathogen wasn't as aggressive. It wouldn't destroy them like a more aggressive potato strain would. It would be more likely then to sprout in the spring, and the symptoms wouldn't be as great. And we're thinking that's also partly why we've had so much trouble finding the sources this spring. But the pathogen starts to develop, it produces spores, the spores then move to tomatoes, and that's the host it really wants to be on, and you see a lot more symptoms.
Okay, is it possible to have late blight guaranteed free tubers?
There's no such thing as guaranteed late blight free tubers. The incidence is pretty low with certified seed, but it is still possible.
Okay, here's a question about disposal, that she's heard a recommendation to dispose of infected plants by bagging and throwing out and putting them in the landfill, but she's concerned because bags usually break when they're picked up by garbage trucks, which seems like a great source of spores.
Very good point, very good comment. That is why we suggest you leave the bags in the sun for a day or 2 or 3. Because inside that black bag with the sun beating on it, it's going to heat up. The plant tissue is going to get destroyed. Remember, this is an obligate pathogen. As soon as the plant tissue dies, the pathogen dies, unless it has produced those resting structures. But those resting structures, those oospores, they're not dispersed by the wind. So in that case, the rotted plants in a bag, if they break open, they don't have spores on them that then can get wind dispersed to a garden.
Okay, we have some questions about the effectiveness of different sprays such as Serenade, compost tea, and biodynamic preparations containing horsetail.
I've seen some efficacy trials that have involved Serenade and Sonata, and they have not been highly effective, and that's under high disease pressure. Copper has been the best. I've seen one trial with compost tea. It was not effective. Compost teas are obviously going to vary batch to batch. I have not seen any work done with biodynamic preparation.
Okay, if rain is a favorable condition for late blight, how do you apply fungicide without it being washed immediately off the plant?
Very good question. Your products that have been developed, such as your coppers, your fungicides, they are developed with adjuvants in them, typically spreaders, what we call spreaders, stickers. They help keep the product on the plant. So if you use a formulated product, they'll have those things in it. It'll help keep it on the plant. Yes, rain's going to wash some of it off, but it's not going to wash all of it off.
Okay, here's some more disposal questions. One is, if UV radiation can destroy spores, is there any way to use UV light to treat plants in the field? And then the second question, is it okay to burn infected plants?
You try to use UV light in the field. I think it would be difficult to reach all the spores. Remember that picture of the leaf where there in that one lesion there was up to 300,000 spores. That's a lot to reach, and they're on the underside of the leaf. So the delivery of the light, while a good idea, delivering it would be difficult. If the pathogen was producing spores on the top side of the leaf, I think it would be a little more feasible. Burning is an excellent way. I had not mentioned it in this presentation because I wasn't thinking of gardeners as considering that as an option. But that certainly is a good option. It's one that some of our organic growers have been using. I think of gardeners as having a smaller, more manageable planting, and therefore would choose to propane flame them. But that is definitely an excellent option.
Okay, here's a question about rotation. How many years for crop rotation if late blight is found?
The reason to rotate would be if the pathogen has produced the sexual structure and it's in the soil. And it's a couple of years. I don't know that off the top of my head. That's something I'm going to need to look up. Otherwise, your source is only going to be those potatoes. So if you're making sure you've harvested them all and any sprouts that come up from potatoes you missed are destroyed, you'll have controlled the pathogen in the soil. But definitely probably a couple years for a rotation. It would be like early blight and septoria leaf spot.
Okay, here's a question about applying copper hydroxide. The viewer got Champ from Johnny, and she said that the application directions were a little unclear, and so she wanted to know if it was critical to cover every leaf stem surface, and what benefit might there be to a quick once-over from above rather than covering every single surface.
A fungicide like a copper is only going to control the pathogen where it lands. So good coverage is indeed critical. Some other products that conventional farmers can use can move into the plant tissue a little bit so then can redistribute. But wherever you can get coverage is important. When I spray my garden, I trellis my plants so they're good and upright, and I can get good coverage. I go in with a sprayer, not just a hand sprayer. I go in with a sprayer that's got some pressure to it, so I can try and get good coverage of the plants. But you also want to be careful that you're not putting too much out there, so you want to read the label carefully and have the correct dose.
Okay, here's a question from a master gardener. I work on a master gardener hotline, and I have a lot of difficulty trying to get gardeners to take samples to a lab because they refuse to pay for anything. So they don't know if it's late blight. And so do you have any suggestions on how to deal with this? We are quickly running out of time, so we're going to have to end soon.
I would check with your local labs. Some labs will provide free of charge a diagnosis for late blight. Lab where I am on Long Island, we will do that. It won't take it any further. So if you want to know what it is, if it's not late blight, you will have to pay for the sample. A good digital picture can be as good as taking a sample in. So if you live a distance from a lab, you could contact the lab and see if they will take a good digital picture and give you a preliminary diagnosis.
Okay, unfortunately we're running out of time. There's so many wonderful questions here, but as I mentioned, if you have any further questions or if your questions didn't get answered, you can go to http://extension.org/ask, and you'll be able to ask the question there as well. I'd like to thank you all for your questions, and I want to repeat also, since we're getting a lot of questions about this, that the webinar will be posted on the eOrganic website at www.extension.org/organic_production, as well as on the eOrganic YouTube channel. So you just go to YouTube and you can type in eOrganic, and our organic farming webinar is already there, and this webinar should be up within the next week to 10 days. Again, we'd also very much appreciate it if you could fill out our follow-up survey, which you'll be receiving in an email. Thank you so much for joining us again, Meg, and thank you all for coming.
Thank you, Alice, and thank you all very much for coming and helping to share information about late blight and its management.